I like to start with an acknowledgement of all the different folks who are involved in this research. This is some of us going down one of the rivers here in Colorado, obviously during the summer, not during the fall, but a number of PhD students and postdocs, a number of collaborators all around the US and some international as well, have been contributing to the work I'm going to be presenting. I'll mention a lot of this work was funded from the CERTIP and ESTCP program, which is a DoD funding mechanism here in the US. So if you're paying attention to these poly and perfluoroalkyl substances in drinking water, if there was a date where things really changed, it was around May of 2016, May 19th. This is the only time I've been quoted in BuzzFeed News was related to this this release of the health advisories, final health advisories for PFOS and PFOA. These are non-regulatory values in the US, but it was two compounds basically saying people should not be drinking water that has more than 70 nanograms per liter of either of these two compounds. This was a national advisory here in the US. Now, what I'll mention is that over the last 5 years, there have been actually regulatory values established by many states, not at the federal level, but some of those advisory levels or regulatory levels are actually quite a bit lower. And in some cases, they're more than an order of magnitude lower. So there are enforceable regulatory levels for some of these compounds in drinking water in the US that are less than 10 nanograms per liter. So really, really low concentrations. People are concerned about these chemicals. So it is a national issue. This is just a snapshot of data that was collected a number of years ago, saying that, you know, of the large water systems, only about 4% had detections, but the key thing to keep in mind is that they were not detecting anything below 40 nanograms per liter. If this study were to be updated, I think it would be much more evident that there's a lot more water systems that are impacted in the US. And indeed, I think this is probably likely the case in other locations around the world. And primarily from my perspective, this is likely due to the use of these aqueous film forming foams, or AFFF materials that are used to put out fuel fires and also in fire training exercises, because these contain very high concentrations, as we'll talk about, of many of these poly and perfluoroalkyl substances. So what are these chemicals? So I'm going to start by talking about perfluoroalkyl acids. And these are fully fluorinated chemicals. They repel both oil and water, used in things like Scotchgard and pizza boxes and clothing. But in actuality, it's the precursors to these perfluoroalkyl acids, polyfluorinated substances, that were used in a lot of these different products. In firefighting foam, it's a little bit different. There's a mixture of both. But the perfluoroalkyl acids themselves are extremely persistent, essentially redefine the meaning of environmental persistence. Some of them are bioaccumulative and can exert toxic effects. So they've been widely detected in people and animals all over the globe. And I think one of the interesting things about this group of chemicals is that we don't usually deal with chemicals that are mobile and bioaccumulative. These chemicals are both mobile and bioaccumulative. At least the ones that are bioaccumulating have a relatively high mobility. So again, what are these compounds? I know there's a lot of chemists on the call here. This is the poster child, perfluorooctane sulfonate, C8F17. So it's got 8 carbons that are fully fluorinated, has this anionic head group. That means that it is a surfactant, and it's actually very strongly retained at the air-water interface, and there's implications of that I'll speak to in a few slides. And the good news from a use in firefighting foam type application is that the carbon-fluorine bond is one of the strongest bonds known. So it won't burn up readily if you coat a fuel fire with these chemicals. The problem is that it's one of the strongest bonds known, which means that these chemicals really are environmentally persistent. And I think they've gotten the name forever chemicals. While there are some folks who think that they've found organisms that can degrade these naturally, I'm myself quite skeptical, and I think that these are chemicals that we're going to be dealing with for decades, if not more, to come. So when I think about the challenges that we're going to be facing with these chemicals, or we are facing with these chemicals, I think about it in a multi-dimensional space. And one of the first things I like to think about is just the compositional complexity of these chemicals. And if you try to rank them with respect to other groups of compounds that many folks have studied over the years, obviously TCE, it's one chemical, right? There is a little bit more complicated chemistry when you look at impurities. But then as we go up this this tower, I would say that pharmaceuticals and personal care products, for those working on those chemicals, are definitely much more diverse in terms of structure because it's really about their function, right? But PFASs are pretty far up there in terms of the complexity of the different types of chemicals that are present. It's not just about PFOS and PFOA. There's a lot of other different chemicals out there. And this is a number of years ago, kind of a family tree effort that was put together by myself and some colleagues in Europe, really trying to lay out what we knew about all these different types of chemicals. And I've highlighted there the precursors as being these precursors to the perfluoroalkyl acids as being one group of chemicals that a lot more information is being gained in recent years, and it'll be a pretty big focus of my talk today. Of course, there's lots of people trying to figure out what these chemicals are, and my hat's off to this this article that was written. It did feature myself, but also lots of other folks trying to identify what these chemicals are. You know, it's kind of neat that the Nature News essentially picked up this sort of work, which is hard in trying to figure out what these chemicals are. So in terms of the first challenge, the way I like to phrase it is, how many different PFASs should we worry about? There's a list in Europe suggesting maybe 5,000 different compounds that are out there. We've actually done some comparison of the Norman Mass Bank list and a list that's now being maintained by NIST, and those don't perfectly overlap. So maybe it's more, maybe it's like 8,000 compounds. I'm not entirely sure. But it's this question: how many of these compounds do we really need to worry about? And I have the paper that a former postdoc, Carrie McDonough, now at SUNY Stony Brook, put together, which really tried to think about how we are analyzing all these different chemicals and where they fit into this bigger space of all the different fluorinated compounds out there. And when we're looking at a typical LC10 mass spec analysis, we're looking for a very targeted list of compounds typically. And that is represented here by this red part of the diagram. But how far out do you need to go to really try and understand this issue? Now, in my own group, we tend to focus on what I refer to as the LC amenable PFASs. So in other words, compounds you can measure relatively easily by high resolution mass spectrometry. And we think that that's a pretty good overlap, not just with the targeted compounds, but also with things that you might assay using something like the total oxidizable precursor assay. So we could spend a lot of time talking about all the other techniques that are out there. This is what I am going to focus on because these are the tools that we primarily use to look at these questions. So if you're not familiar with the TOP assay, my hat's off to Erica Houts and David Sedlak at UC Berkeley for developing this. The idea was, if you take all these polyfluoridated compounds that might transform to perfluoroalkyl acids over time, you subject them to high pH conditions to generate hydroxyl radical, you convert them to the measurable perfluoroalkyl acids. And it's not an even conversion. There's some chain shortening for some of these compounds. But what it enables you to do is if you don't know what the parents are, you can at least measure what the products are. And so this is an example where Erica applied this to different firefighting foams that had been used in the US in different ages. Note the scale here is grams per liter. When those same foams were subjected to the TOP assay, what we ended up with are grams per liter concentrations of several of these different perfluoroalkyl acids, carboxylates all shown here. And so this is one tool you can use to try and study the chemicals out there, is essentially inferring their presence via this TOP assay. We prefer to look at HRMS analysis. So this is using high resolution mass spectrometry, primarily QTOF, but we do have an Orbitrap now in my lab. What we're looking at here is a large diversity of structures. And It's quite it's a tongue twister, I should say, to try and pronounce any of these names. If I were there in person, I'd offer a beer to anyone who could give me a good pronunciation for example, this compound here. But the point is, there's lots of different compounds out there, and most of them we can analyze by high resolution mass spectrometry. So when we go out to look at an HRMS impacted site, we're looking on the order of 1,400 different compounds. We don't necessarily see all of those, but that's what we're looking for, because these chemicals all have different head groups shown here. So the head group, not just a sulfonate, can be a sulfonamide, a fluorotelomere, amide of some sort, but also different chain lengths, so different short and long chain compounds. So when we go out and look at sites, we often see tens, if not hundreds, of different compounds. So one of the things that I wanted to really focus on today is not just that first question in terms of composition, but really the second question, the second dimension I like to think about, which is the environmental behavior. And this is where I think PFASs really start to pull ahead of a lot of the different compounds in terms of their complex behavior. And of course, we could debate where all the other compounds classes fall in there. But from my perspective, this is where PFASs really start to differentiate themselves, particularly because of that the difference in chemical composition. So it's related to those structures, how they're behaving in the environment. I'm going to focus in here on chemicals that were used in the firefighting foams. So this is the aqueous film forming foam. It's important to note that approximately only 3% of fluorochemical production was or is for aqueous film forming foam. A lot of that was used by the military. The 75-25 is somewhat debated. I've heard 50-50 in other areas. And so, you know, these materials were used to put out fuel fires. They're complex proprietary mixtures. Jennifer Field's done a lot of work to try and identify the individual components in there. And the point is that when these compounds are used at these firefighting foam impacted sites, they're often directly released. So you spray it on the ground, or it was sprayed on the ground. And the concentrations are really high. So you saw from that top assay slide is that grams per liter levels of either poly or perfluoroalkyl substances can be present in some of these formulations. Just to give you a sense, if you had a bucket of 3M light water, one 5-gallon bucket, I realize gallons is a little bit tricky for the Europeans, but if you had one 5-gallon bucket of this historical 3M light water, it has enough PFOS in it to contaminate a annual supply of water at 70 nanograms per liter for a town of 27,000 people. So that's just one bucket, and we've got some of these buckets actually in our lab. So it's really, really highly concentrated. The other way to think about it, you know, people say, well, how many 7 nanograms per liter, 70 nanograms per liter is, you know, how many drops of salt in an Olympic-sized swimming pool? I like to flip that around and say that if you had one Olympic-sized swimming pool filled with this light water, it would have enough PFOS in it to contaminate 7 years worth of the water supply for the entire United States. And so it's a lot of water that could be contaminated. And we made many, many Olympic-sized swimming pools of this material over the years. So what this means is that, you know, we looked at the earlier studies suggesting where things have been detected. This is just the list of potential known and potential release sites in the US. It's a lot more when we think about that local fire stations, for example, which are not even shown here, that have volunteer firefighters. Those are also use these materials, and they're not shown on this map. So there's a lot of places where these foams could have been released due to usage not just at military facilities, but also in small towns. So the question that I want to focus on, went a little too quick there, is why are they still in the soil if these chemicals have been released at the firefighting foam sites? Or the other way to think of it, hold on, is if these compounds are so mobile, why are they still in the vadosome soils, or the soils right at the firefighting foam impacted sites? And I won't get into the details there in terms of the data that back that up, but when you go out and look at some of the soils at these sites, they're often very, very, very highly concentrated. And some estimates are that 90% of the mass of some of these chemicals is still in the soils at the sites. And so the working hypothesis as to why this is, given the fact that they are so mobile, is that there's two reasons why they might be retained in soils. One is that they have air-water interfacial surfactant properties and therefore are retained in unsaturated soils where you have a lot of air-water interfaces. And the second is recognizing the role of precursors. And the idea here is that there can be potentially slow release and biotransformation, presumably biotransformation, of some of these cationic and zwitterionic precursors. And I didn't point that out on that slide when I looked at all the different structures, but if you were paying attention, there weren't just anionic moieties on those molecules. They were also cationic moieties, which could contribute to the a stronger retention of these compounds in soils. So we actually are doing some work on this air-water interfacial transport. This is actually not from our study. This is a study from Marc Brousseau. But the short answer is because these chemicals are air-water interface surfactants, decreased soil water leads to increased retention because they stick at that air-water interface. And that's shown here, the breakthrough curve of the saturated soil for PFOA in the saturated soil versus the unsaturated soil. It slows down the transport and therefore the release of these chemicals because of that air-water interface. So this is certainly one thing that is bearing out in the literature, that these chemicals are retained at many air-water interfaces, and that is contributing partially to the retention in soils as compared to movement to groundwater. What I'm going to focus on, though, is this issue of the precursors present in soils and slowly being leached and transforming potentially to the perfluoroalkyl acids. So the questions really there are, are these precursors abundant in HLF impacted soils? Are they more slowly released as compared to the perfluoroalkyl acids? And will they transform to the perfluoroalkyl acids? So again, what I'm talking about here is precursors such as this particular chemical is actually not used in the firefighting foams. But if this chemical slowly transforms over time, it would lead to elevated levels of PFOS in the soil and then into the groundwater. So to answer some of these questions, I had another former postdoc who is now working at NIST, Andrew Meisel, and this paper actually just got accepted on Friday, so be on the lookout for it coming out soon. But he wanted to look at how these chemicals are released from these HLF impacted soils, particularly focusing on these precursors. And so he set up some column studies where we pumped artificial groundwater through some soil that was collected from the field, and then analyzed both the soil initially and then the effluent coming off that soil for everything we could think of that was amenable to LC high resolution mass specs. We used, in this case, LC QTOF-MS, doing both target analysis, suspect screening, and a little bit of non-target analysis. In the end, we actually didn't see anything in a true non-target approach, so we really focused in on the suspects. For those who, I know there are folks who care about how we did this, so I did throw this slide in here. We essentially used two different approaches where we had a match for our accurate mass match, where we looked at the parent mass and looked at the isotope patterns and said, does this match a suspect on our list? But then we also, because we had built up this library of MS2 data for all the different foams that we looked at, we were actually able to look at the MS2 data for a number of compounds and essentially get library matches as well. So again, we don't have authentic standards for a lot of these compounds, but we can get varying levels of confidence in terms of our identification of these compounds in our systems. So before I get into what Andrew showed in his soil columns, I want to point out that a lot of it relied on some excellent work from a now former PhD student, Anastasia Nickerson. And what we realized is that if we're talking about these cations and zwitterions in soil, our standard extraction procedure, which is a pretty much a basic methanol procedure, wasn't going to get everything off the soil. And so Anastasia took it upon herself to develop a new method to enhance the extraction of these cationic and zwitterionic compounds. So this just gives you a sense of it, where we had a basic extraction, and we followed with some acidic methanolic extraction, a cleanup. Obviously, you had to neutralize the extracts before combining and then essentially evaluating on our LCQ to FMS, using as many internal standards as we could and trying to quantify things as best we can for the target analysis. But there is this question as to how we estimate the concentrations for the compounds that we don't have standards for. But again, I'll note here we are doing it both ESI positive and ESI negative mode. This is where Andrew, also working with Anastasia, developed a series of R scripts. It's not that complicated, where we could essentially estimate the concentration of a lot of the compounds related to response factor for the different compounds that we had standards for that shared similar ionizable groups. So this is an example of where we had a substituted, for example, derivative of a perfluoroalkyl acid, and we were able to estimate its concentration using PFOA in the internal standard response for PFOA. So the idea here is it's not perfect. We realize these are semi-quantitative numbers, but it gives us a sense of how important some of these compounds may be. And when we apply this sort of approach to soils, and this is an example of Anastasia's work showing the concentrations of the different precursors in soils, as well as some of the targeted compounds. What I've shown here is on the left, shows 3 different soils, and on the left is the ratio of the what we saw in our enhanced extraction procedure versus what was observed in the basic only method, the old soil method. And on the right, the scale is our semi-quantitative or quantitative concentration, and they're ranked from left to right in terms of concentration. And what's important here is that the perfluoroalkyl acids, such as PFOS, were not always the highest in concentration, or at least our estimated concentrations, recognizing that there's some uncertainty around those semi-quantitative concentrations, and that a lot of these compounds, some of the higher concentrations are actually the ESI positive compounds. So the cations and zwitterions are actually really important in some of these soils. And I should mention all of these soils were extracted from, were collected from source zones here in the US. So the enhanced extraction procedure does get more of these compounds out. And because we're looking in a full scan mode and both positive and negative mode ionization, we're able to see a lot more chemicals present in these source zone soils than people had initially thought. One of the interesting things, and this will maybe be time for discussion for later, is when we do compare our semi-quantitative approach versus the top assay, in the first soil we looked at, it actually matched up relatively well. So what we observed via target and semi-quant and compare that to the top assay, things were pretty consistent. However, when we looked at the other soils, it wasn't quite as good. And so somewhat surprisingly, our semi-quantitative numbers were more than the top assay, which gives you suspicion that maybe the semi-quantitative approach wasn't, we were off by a significant factor. But one of the interesting things is that during the process of developing this paper, some of these compounds actually became available as standards. And those compounds that we can compare against, our estimates were actually pretty darn good. And so we don't think this is solely just because the semi-quantitative approach is wrong. There may be something else going on here in terms of the top assay capturing all the different compounds that we can measure via HRMS. So going back to Andrew's column, so now that we have a soil method, we can look at these compounds, we can semi-quantify them. What do we see coming off these soils when we pass water through these soils? Well, just to give you a sense, you observed 167 different compounds from these 2 different soils. And if you look, the red ones are all precursor compounds. So these are lots of different compounds that could form these perfluoroalkyl acids over time. But what was really interesting is that the release profiles of these chemicals are very different. So what we're showing here on the top is the release profiles. So C over C max, so the relative, the maximum concentration for each individual chemical, and the concentration via pore with respect to pore volume. And note the pore volume is log scale here, where we see the carboxyls and sulfonates, which are the top 2 panels. Those are showing pretty straightforward chain length dependent elution. In other words, the short chain compounds come off the soil first, the long chain compounds take a little bit longer to come off. So generally that fits with everything we know about the behavior of these chemicals. But what's interesting is when we look down here at the bottom is that when we look at, in one case, one subclass where all we're changing is the perfluoroalkyl tail length, we do see a little bit of that chain length dependent effects. That's essentially what we're looking at over here. So this is the C3, C4, C5, C6. We see them eluting in that order, but you'll also note they're coming off much, much, much later than the other compounds up above. And that's best illustrated over here, where instead of holding the head group constant, we hold the perfluoroalkyl tail constant. So PFHXS, this sulfonate, which is the slightly reduced version of the sulfonate, sulfonamide, and these other types of precursors, there's big differences. That head group is a major factor in terms of its retention in the soil. And some of these compounds aren't really coming off until 100 pore volumes have passed through the soil. And I should mention, most of those compounds down there are cationic or zwitterionic compounds. And this is really evident when you look at the fraction of the mass eluted for the top 10 compounds in these two different soils. In the case of the blue soil, one of the soils, we actually saw a lot of the compounds coming off, except for these precursors down here. And in the other soil, where we saw a lot of these precursors that actually were in the top 10, a lot of them didn't ever come off. So even after 100 pore volumes, a lot of that mass still hadn't eluted. And it's really important that when you look at the relative mass, even if the precursors that do elute, if we think about this as PFHXS, as well as anything that can form PFHXS in the soil, this sulfonamide and this other sulfonamide precursor swamp the mass of the PFHXS there. In other words, there's much more of these precursors present specific to PFHXS that are taking a lot longer to come off the soil than the PFHXS is. And so this really speaks to the fact that the precursors are present and can meaningfully contribute to the load of PFASs coming off of the soil. And I'll particularly draw your attention to this C6 sulfonamide, FHXSA, sorry, because you're going to see that becomes important when we look at some of these other sites. But before I do that, I'll just briefly say this is entirely consistent with what Anastasia also saw in a field site. This is another paper where we did high spatial resolution sampling analysis of soil and water. This is the direction of groundwater flow in our fire training pit where the chemicals were released. And we took a transect here and looked at the concentrations in the soil and groundwater. This is in conjunction with Jennifer Field's group and folks at GSI Environmental. And if we look at the total concentrations of the PFASs, this is from left to right is the soil core profiles and the groundwater profiles, again, from left to right as a function of depth. And what you see is we see really high concentrations in the soil, in the source zone. In the groundwater, things are moving down gradient, which makes sense. We know these plumes develop, but the concentration is much more even in the groundwater than it is in the soil. There's very much a spatial difference here for the soil in terms of total PFAS versus what's in the groundwater. What's really interesting is that if we look at just the percent that are attributed to the cations or zwitterions, again, recognizing that we're semi-quantifying here for a lot of these compounds, so it may not be perfect, but a very, very large fraction, 97% in some cases, of these concentrations shown on the left are attributable to these cations and zwitterions, whereas down gradient is really minimal. So a lot of these compounds are really sticking in that source zone where they were initially applied. Maybe some of them are migrating down a little bit in this particular site, but they're really not moving nearly as much, which is entirely consistent with what we saw in the soil columns as compared to the other compounds such as the perfluoroalkyl acids. So the last kind of perspective with respect to complexity that I want to give you, the last dimension, is this question related to human exposure. And that's where I really think that PFASs really kind of are important and are extremely complex because it's not just the different types of where you're exposed to, but the different forms of the chemicals that might be present upon exposure. So this is a nice graphic put together by Elsie Sunderland, which I modified slightly, that talks about human exposure to these compounds. So we can be exposed directly from consumer products. We can be exposed from the environment. There's all sorts of ways that the chemicals are released to the environment. We talked a lot about this firefighting foam sites, and I'll continue to talk about that. But the point is, at the end of the day, we're really concerned about how people are being exposed, and animals, but primarily people are exposed to these chemicals. So the third question is, from my perspective, the third challenge, particularly with respect to firefighting foam sites, is which of these compounds should we worry about for these firefighting foam impacted sites, of which there are thousands, and specifically, which ones should we be concerned about impacting drinking water supplies? So people have measured PFOS and PFOA and a few of the other compounds, but are there a lot of other things that we need to think about? And so I break this question up into kind of three things. One is which compounds are there. The second question is which compounds might bioaccumulate. And the third question is which compounds might pose a hazard. The working hypothesis is that there's a lot of different compounds that might be released, mainly the shorter chain compounds and perhaps shorter chain anionic polyfluorinated substances might be important in these AFFF-impacted water supplies. We think if they polyfluorinated substances are there, they're probably less bioaccumulative than perfluoroalkyl acids. And the third question, which compounds pose hazards? It's not really an exposure question. I'm going to leave that one to any toxicologists out there working on this question. So what we're really getting at is this question of, do these polyfluorinated substances matter when you get all the way down to an AFFF-impacted water supply, recognizing most people aren't drinking water that's coming from a firefighting foam impacted source zone. They're drinking water that's far down gradient. So one of the first indications that it's not just PFOS and PFOA that people might be exposed to came from a paper that a visiting student from China, Zhen Zhao, worked on with me here at Mines, where we looked at different compounds that might be present in water, and then we tried to evaluate how well they would be removed in an activated carbon type setting, which is still relatively the dominant way that many people remove these compounds from water, although I'll say that anion exchange resins are also gaining a lot of popularity. So in this particular water supply, he observed 30 different compounds using HRMS and or authentic standards. And I'll want to point you to the fact that he did see these sulfonamides, these compounds that actually constitute a large fraction of the mass that come off much later from our soils in the soil systems. I will mention that he didn't see any cationic compounds, and most of them were anionic. So there were some zwitterions that were being transported. This particular site, we think, had some fractured bedrock, which might have facilitated transport of zwitterionic compounds. But the point is that we did see a lot more compounds than PFOS or PFOA. This was followed up by a study here in Colorado by Carrie McDonough, where she looked at water impacted in the Security-Fountain-Widefi district, water districts in just south of Colorado Springs. And this just gives you a sense of the levels of, in this case, we didn't have standards for a lot of these compounds at the time, the relative concentrations or peak areas of these sulfonamides. So these are, again, this is the structure of that FHXSA, and we see that in a lot of the water samples from these well fields that these water systems were using. What's interesting is that they're not necessarily correlated with the perfluoroalkyl acid. So this shows the perfluoroalkyl acid data, and we saw some of these wells actually had elevated levels of these sulfonamide precursors, but some of them did not. So that poses an interesting question as to the dynamics of these chemicals moving through a groundwater system. At the end of the day, though, what about bioaccumulation? Do we care about these compounds with respect to bioaccumulation? And this is where Carrie partnered with Jamie DeWitt, who actually decided, well, why don't we expose mice to HFF and see what actually accumulates? And she was also looking at toxicological endpoints. We're not going to talk about those today. But what Jamie did, she did the exposures where they dosed mice with HFF. And then Carey was looking in the serum and said, okay, what do we actually see in the serum as compared to what was in the firefighting foam? This is an analogy of a firefighter maybe directly ingesting accidentally some firefighting foam if they were using it, but also trying to get a sense of, do any of these precursors potentially transform in vivo to the compounds that we're more concerned about? This is a sense of what we observed in the HFF. So this is a bubble plot, which gives you a sense of the relative size of the bubble is relative peak area, and then the mass to charge ratio versus retention time in the system. So what you can see here is there's a lot of these sulfonamide precursors here in this 3M light water HFF. Yes, there's a lot of PFOS and other sorts of compounds. We actually took that off, so you weren't necessarily seeing that here. But when you get into serum, you don't see nearly as many compounds. In fact, very few of these sulfonamide precursor chemicals were actually observed in the serum of the mice that were being exposed. So the precursors, this was suggestive of the precursors not accumulating in serum. But that doesn't get at the question of were the mice transforming some of these precursors to the more problematic or potentially more problematic perfluoroalkyl acids in vivo. And so to get at that, we devised a way to think about this in terms of bioaccumulation factors. So we call it a pseudo BAF because it's not exactly a BAF. What we did is took the peak area of the chemicals we observed in the serum, normalized the amount of linear PFOS we saw in the serum, and then we compared that against the peak area in the HFF spiked blank serum, normalized again to the linear PFOS. So the idea here is to try and get rid of any matrix effects that might be affecting the response by having both things compared in serum, but then also because we don't know the absolute concentration of some of these things, at least compare it to a benchmark chemical, in this case, linear PFOS. And what's interesting is that when we looked at both the male and female mice, although there's some differences between them, is we see relative to the PFOS, some of PFHXS and PFHPS suggested they're actually more bioaccumulative, but we know that when you do exposures with some of these compounds directly, they're actually less bioaccumulative. So that to us suggests that there's likely precursor transformation leading to these elevated levels of these compounds in the mice. And this holds for some of the other compounds too. So the C9 sulfonate and PFOA as well. Over here, what is interesting is that it's not just about the end product chemicals accumulating. Some of these substituted PFOS-like molecules which we don't necessarily think there were precursors for, they themselves might be actually slightly more bioaccumulative than PFOS. Now, keep in mind, their concentrations are likely a lot lower. In fact, we estimate concentrations much lower than PFOS in the foam, but some of them might be actually more bioaccumulative. And so you have to take that into account if you were to do any sort of risk assessment for some of these compounds. So taking a step back, if I think about the challenges for environmental engineering and the public health community with respect to PFASs, I think the compositional chemical complexity is daunting, but it's a tractable challenge. HRMS is becoming much more common. There's a lot of folks who are helping advance this approach by providing lists of compounds. There's a development of MS2 libraries, which would be platform independent that people can use to compare against. And I think we're going to have a much better sense of the different types of chemicals that are out there as more and more of those folks are using these tools. I will say that the transport of these compounds and their fate in soils is complex, but most of it is entirely consistent with known environmental physicochemical phenomena. So, it's not that you have to throw out the FOC, KOC paradigm. That actually will go a long way, but it's not sufficient. There are other things happening for these compounds. The precursors are strongly retained. They might be transformed. There's air-water interfacial partitioning happening. So it's a complex, lots of complex things happening, but again, it's relatively tractable, and they're consistent with what we know if we just go back and look at all the data that's been collected over the last 30 some odd years of chemical behavior in the environment. With respect to the human exposure question, I do think that precursors are likely present in many, if not all, AFFF-impacted drinking water supplies, and they're likely contributing to PFAS body burdens. And so I will note that if you're treating the water to remove the perfluoroalkyl acids, you're likely going to be treating these chemicals and removing them as well, but they can potentially be result in exposure pathways, maybe perhaps via inhalation if you're only treating the drinking water and people are still showering with this water. There are potential other exposure pathways that we might need to consider given that the precursors are there. And also, what does this mean from a toxicological perspective? These precursors tend to be more reactive, and therefore is that more problematic from a tox perspective? So if we look at this kind of iceberg perspective, how should we communicate some of these unknowns about these chemicals potentially present in our water supplies? And I think that's a good question, except if you're paying attention to social media nowadays, it really becomes a question of how we should treat for the unknowns. And actually, some people say, should we treat for the unknowns? And I say, when you have these sorts of communities that have kids, in these cases, this is where they have very, very high levels. These are nanograms per mil of PFOA in their blood. It's not a question of how, it's it's a question of how and what sorts of technologies we should be using to remove these chemicals from our water supplies. With that, I'm happy to take any questions. My lab is shown here. You're kind of getting a view out the back here, but I'm happy to answer any questions that folks have.